Hi, good evening, everyone. Hello.
Good evening, teacher.
Yeah, good evening. Uh, is that Saindi?
Yes, teacher.
Yes. Is our other friend in fast? Is that?
Yes, teacher.
Yeah, good evening.
Good evening, teacher.
Yeah. So today we are going to start a new class. So first of all, I'm going to give you a little introduction about me. So I'm a new science teacher for your grade 9 class. Then today I'm going to tell you about me at first. So I'm Sandamali Jayakody. So I'm a graduate of University of Sri Jayawardhanapura, and uh, can you hear me well? Can you hear me well?
Yes, teacher.
Yeah, okay, it's fine. So after completing my degree, I'm conducting classes for grade 6 to 11 students for their O level at the end. So now you have connected with us to get ready for your new science lessons in grade 9. At the end of the grade 9, we are moving to O level classes at the end. So, first of all, um, that's about me. Can you tell me something about you all, Savindi? What about you? What's your school?
I am Savini Ravanjana. My school is Senance Balika Mahavidyalaya.
Yeah, good. And in first, what about you?
I am Infa Sudin. I am going to Zaira College. I
Yeah,
am 14 years old.
14 years. Yeah, good. And we have new friend. So Dean, can you hear us? Yeah, he's connecting.
Teacher, I am in fact teacher. This is my phone because I had a network problem. That's why.
Ah, yeah, it's okay. So I thought is there's another one. Okay, it's fine. Uh, so, uh, so shall we move to the lessons? Before that, I have to tell you something. Are you familiar with this platform? That means the website. Is this really fine for you, or shall I show you what do you have, what you can do with that? Shall I show you at first?
Yeah, sure.
Okay, so I'm going to show it. Then I'm going to share my screen with you. So this is the website, and here you can join with as a student. Here, this is my profile as a teacher, and in this website, at that point you can see login, and then you can log into your profile. After that, you can download the PDF tudes I have sent you. Did you collect it?
No, teacher.
Uh,
No,
there
teacher.
is one. This is the tutorial I have sent you. Grade 9, lesson 1. Didn't you see that? There's a wrong one. It's grade 9 one. This is the tute. Grade 9, lesson 1. You can download it with that options you have from your side. And then later on you can do it. Okay.
Okay, teacher.
Okay. There are some spaces in that tube to fill while I am doing the class. But since you are not having that tube with you, so you can interact with me and you can discuss the lesson with me. Later, you can fill all the things in it after completing the lesson. And then this is the part which you can answer to the some MCQ sessions. There may be 5 or 10 MCQs. You can answer these things and you can resend those things. Then I can check it and give you a feedback about those things. So this section, it's about papers. Maybe a monthly paper or maybe a paper which is relevant to your lesson. It is uploaded to this part. You can download it and then you can upload the answers. So that is the normal process in your class. And then we have another friend. Give me a minute. Yeah. Is this Iron? Hello, good evening.
Hello.
Yeah, can you hear us?
Yes.
Yes, okay. So I was showing the way to get the tutorials and way to answer, way to upload your answers and all the things. Uh, I'll show you that again. Okay. So here we are going to start the class now. So our first lesson today is the application of microorganisms. So before starting the lesson, shall we have a little chat about microorganisms? So
Teacher
here,
can't.
you can see it. Yeah, okay. Share screen. And then fine. Can you see now? Can you see the slides now?
Yes, teacher.
Yes, okay. So today's lesson is about applications of microorganisms. So do you have any idea about microorganisms? How do they like? Do you have any idea? Why
They
we
cannot be seen clearly with the naked eye.
Yeah, that is the main feature. That is the main feature of these microorganisms. They cannot be seen with naked eye. Really good. Micro means that they are good. They are really tiny. They are really tiny organisms. So we are going to discuss about these organisms today. Not only about their features, we are going to discuss how do they important for us. What are the importances? What are the what are the advantages we are taking from them? We are using them in different ways in our day-to-day activities. That means for our day-to-day users and also for different industries and different medical applications. We are going to discuss all of these things today. So the first thing we are going to learn about their features. Features of microorganisms. Here, as you all said, they are microscopic. They cannot be observed by naked eye. Here you can see there are some strawberries and there's nothing in it and it's just an illustration. But once we are using this lens, we can see them. But actually we can't see them with this type of hand lens, but it's just an illustration. It's give the idea that they are microscopic. We can't see them. So we can use, it's like compound microscope to see them. So that is the main feature of these microorganisms. And then in next feature, they are unicellular or multicellular. What is the meaning of unicellular? It is given here. Can you read it? What is the number of cells they are made up of? Unicellular. That means they are made up of. Can you complete my sentence? They are made up of?
Cells.
Yeah, made up of cell. And here it's a single cell. Unicellular means a single cell. They are made up of a single cell. If not, sometimes there are some microorganisms who are made up of few cells. There may be 2 cells, 3 cells, and more than that, maybe few cells. So we call them as multicellular. They have more than 2 cells, two or more cells. So in that way, that is another feature of them. And then next feature is they are growing fast. Once there's a one microorganism, it grows fast and spread each and everywhere. As an example, normally in laboratories, microorganisms are growing by just using a single drop of a microorganism sample. But it grows entire everywhere within a short period. They grow fast because they have a high rate of reproduction. The meaning of reproduction is they are producing new microorganisms, new microbial particles at a high rate. And then there's another feature, another important feature of them. They are widespread in nature due to their ability to adapt to different environments. They can adapt to different environments very well. That means they can live everywhere. They can live in everywhere. Like one example is they can live in our food. They are living on our food, or they are living in our food. Sometimes they are living in our body too. If we are having a disease, they are growing in our digestive tract. Once we are having a digestive tract disease, normally, not only in situations like diseases, they are normally growing. We call them as favorable microorganisms. They are just friendly microorganisms with us. So they are also living in our body. Another one student is coming, Vinod. Hi, Vinod. Hi, Vinod, can you hear us? Vinod, can you hear you? Can you hear us?
Teacher,
Yeah, we can hear. How are you, Vinod?
I'm good.
Okay, okay. So we started a little bit. Shall I give you just a little introduction again within few seconds?
Okay,
Otherwise,
okay.
since this is the beginning, otherwise it's not fair to continue
Okay,
from
teacher.
here. Okay. So we will started to discuss about microorganisms.
Okay, okay.
So this is in this lesson, Vinuja, we are discussing about the applications of microorganisms. So we
Okay.
started first looking at their features. So this is the thing. We discussed that they are microscopic. That means they are tiny. We cannot be seen by our naked eye. And also they are unicellular or
Okay.
multicellular. That means single cell or made up of 2 or more cells. Did you understand?
Okay. Yes,
Okay.
yes.
Yes. And then they grow fast. That means they have a high rate of reproduction. One
Okay.
microorganism, maybe a one bacteria, become 2, and then they become 4. In that way, they multiply very fast. And then we discuss they are widespread in nature due to their ability to adapt to different environment. They can adapt different environment, and they live very well in each and every environment. That means they live in our body, they live on our food, and then they live each and everywhere, even different extreme and in different extreme environment they are living. Later on, we are going to discuss about what are their environments and what are the substrate they are growing on. Substrate means the place, the surfaces where they are growing on. So now all of we are going to discuss about these different microorganisms. There are few different groups, but before that I have to tell you some different environments they are living. When we talk about their different environments, they live in water, they live in air, they live in soil. So each and everywhere they can be found. Sometimes we are talking that water is polluted with unfavorable microorganisms. That means disease causing bacteria and disease causing some different microorganisms are growing, are spread on that water. In the same way, soil is enriched with microorganisms. There are enormous amount of different microorganisms in soil. And atmosphere is also same. There are many microorganisms in atmosphere too. That means in air, they are floating. Tiny microorganisms are there. So we are getting some diseases due to this airborne. That means the microorganisms who live in air. So they adapt to live in those environment. And I told you that they are living in extreme environment. So extreme environment means, extreme environment means they are living in different places where we cannot live. That means it's like desert. Maybe it's a really difficult place for us to live. And then hot water springs, they are living in these things, but it is not easy for another different organisms to live in those environments. Hot water springs. So these are the extreme environment they are living in. Hot water springs. So that's all. Now we are having a little overall idea about these microorganisms. Do you have a clear idea about these microorganisms now? How they like?
Yes, teacher.
Yeah. Now you have a rough idea. Now we are going to discuss them in detail. So these are the main 5 microorganism groups we are going to discuss today. They are bacteria, protozoa, viruses, and fungi and algae. So these are the main 5 groups we are going to learn. This is the first group, bacteria. So we are going to learn about the features of these microorganisms. Here it is mentioned that they are unicellular. That means they have a one cell. Only one cell is in their body. That means just a single cell. Then different body shapes. They can be round and they can be like this, rod shape, round, then is circular or rod shape, or this kind of different shapes. There may be different shapes of the,
Excuse me, teacher.
yeah.
Do I have to write a note today?
Uh, so I have uploaded a PDF tude before the class. So later on, you can take a printout of that and put the things in that, because there are some spaces for you to fill in that. Normally, for at the beginning of each class, you have to have that PDF tude as a hard copy on your hand. But today, I think any of you don't have that with you, since this is the first day, isn't it?
Yes, teacher.
Yes. Is anyone having?
No, teacher.
No, not anyone. So it's fine for today, but later on you can take the printout and ready for the class when it is going to start. So today, due to that reason, we don't have anything to write. You can discuss with me and you can interfere with me, and later on you can fill the spaces, because this video is going to be uploaded for a little period onto that platform. Then you can refer it again, then you can answer, you can fill the tude. Okay, is that fine?
Yes, teacher.
Okay,
Yeah.
teacher.
Okay, for today don't mind it. Later on you can do it. This is, since this is the first day, it is the problem, but from further classes you can do that. Then these are the features of bacteria. The third feature is they are widely spread in nature. They are widely spread here and there, everywhere they are widely spread. So here are some examples for this bacteria. In milk products, we have a special bacterium to produce yogurt and to produce different dairy products. We are using that bacterium. That is, the name of that bacterium is Lactobacillus bulgaricus. Lactobacillus. You actually, in this lesson, you don't have to remember those names by heart, but you can just have an idea on your mind. It is Lactobacillus. Normally, we have an idea that milk is just lactose, and even we are having some milk products with lactose and this and everything. So you can remember that name easily. It's a lactobacillus bacteria, which is in milk products. That means dairy products. And then this is anthrax disease. It is causing by a bacterium. Here you can see this blue color rod shaped things. Can you see?
Yes, teacher.
Yeah, they are the bacterium. They are the bacterium which is causing anthrax disease. We call it as Bacillus anthracis. Here these round cells are normal blood cells in our body. This is just a actual image of an actual sample. Blue color things are the they are colored bacterial particles, and round big objects are our blood cells. Here you can see the sample has taken from the wound or the lesion on a hand. There's a big wound caused by this bacterium. That means our outer skin is infected with this anthrax bacterium. Anthracis bacterium. It's a bacillus bacterium. That means it is just rod shaped like this, as in the same way like this lactobacillus bacteria. So it is another example here. Cholera disease. It is caused by a bacterium like this. It is rod shaped like this and it has a nice tail like this. So we call it as a filament. This tail is a significant feature of this bacterium. This gives Vibrio. This is the name of this one is Vibrio cholera, and it makes the cholera disease in us. And then so these are some anthrax disease, cholera disease. These are some advantages for us, disadvantages for us. But when they are in milk products, when they are in vinegar production, these are important. Vinegar is produced from coconut sap, also from different plant fluids. We can produce vinegar. There are different sources. Even from coconut water, we can produce vinegar in home. So vinegar production can be facilitated by normal bacteria who are available in natural environment. The name of the bacterium is Acetobacter. Acetobacter makes vinegar. That word reminds us acid, Acetobacter. Acid means vinegar is just acid. It is having a very very strong flavor. So it's just like acidic flavor. So it is done by Acetobacter. So these are the normal information about bacteria. And then we are moving to discuss about our next group, fungi. Give me a second, just like stop. Oh, it's going back. Yeah, here it's fungi. Here on now it's unicellular or multicellular, not just unicellular. There are some examples who are multicellular, made up of few cells. Now I am drawing few cells of the multicellular virus, so multicellular fungi. This is a unicellular fungi, and here clear image is given. It is named as yeast. Yeast is used in bakery industry. And then its name is Saccharomyces. Saccharomyces. Yeast, yeast is the normal name of that fungi. It has a scientific name too, Saccharomyces. No need to remember the scientific names very well. I'm just mentioning. Just remember it is yeast. It is a single cell, unicellular. And not only unicellular ones, there are multicellular fungi. Just as an example, fungi on bread. The name of this fungi on bread is mucor. Hope you have a little exposure for these microorganisms in your first lesson in grade 8, isn't it? Can you remember?
Yes, teacher.
Yeah, you have a little idea. You were learning about the food spoilage and all the thing. You had to keep a piece of bread and observe the microorganism growing on it. So that is mucor. Normally we can't see with this naked eye because they are microscopic, but we can see just little black color spots on bread. So that is mucor. And although they are microscopic, we can see their reproductive organs sometimes. So this is their reproductive organ. Normally we call them as mushrooms, but actually that is the reproductive organ of this type of this type of fungi. So they may be like a filament. When you are looking at a place where some dry leaves are collected and they are decaying, you can see some white color strips like things. Have you seen ever? White color small strips on a garbage pit or somewhere when there's a decaying wood log, you can see that. Have you seen that? Little white color strips on the dead leaves like that. Have you seen ever?
No, teacher.
No. So, okay. I try to show it. Fungi. Just like this. On decaying wood logs, on decaying dead leaves, you can see this kind of small white color strips on that. Can you see this image?
Yes, teacher.
Yeah, these are named as fungal mycelium. Mycelium means just little filaments. We can see white color strips on decaying woods or decaying leaves. So those can be observed by naked eye. And also at the end of their life cycle, we can see nice mushrooms like this. So they are the reproductive structure of these fungi. Then at the end of this, at the end of the formation of this mushroom, they are forming little spores on it, and then they are falling on the ground again. Then they are again forming the new fungi filament. So that is how they are reproducing. So you have to remember that although fungi is microscopic, their reproductive organs are visible to naked eye. What is the name of that?
Mushroom.
Mushrooms. That is the reproductive structure of fungi. And then they can thrive on moist surfaces. Moist surfaces means normally we can use this word too. For the moist surface mean, for the surface we are using substrate. We can use this word too. Substrate. Substrate. That is the surfaces there where they are living on. Normally we can't see they are on very hot places, very dry places. Normally they are in very, very moist phases like garbage pits and below and under the trees in that way. So that is about fungi. Now we are moving to discuss about protozoa. This is a special group. They have a special ability to move like other organisms, other normal animals we know. They can just do movements like swimming like movements. So we can call that they can locomote. They can go here and there. The first one given here is a amoeba. It's a unicellular one. Normally this image also amoeba, and this is a real image of amoeba. You can see here it is just a single cell, but its parts of that cell are moving here and there, just like legs. They are moving here and there. So we call this little leg like hand-like projections as pseudopodia. Pseudopodia. So it's an appendage which help them to locomote from to move from one place to another. And here this one is paramecium. It has a nice structure like a it's like a shape of a shoe, bottom of a shoe. So it has little projections like this around it. We call these projections as cilia. Cilia. And then that is paramecium. Now we have another one here. It is euglena. Here you can see a clear image of euglena. This is a real image. It has a little tail-like structure. This tail-like structure is named as flagella. So can you tell me again, what are the appendages in protozoans for their locomotion? There are three. Can you just read it loud? Can you all?
Cilia,
Yeah,
pseudopodia, flagella.
flagella. And what about my other students? Can you just read it loudly? So you have to discuss with me today since you don't have nothing to write here today. Okay. Ayan, are you listening well?
Yes.
Yeah. Okay. So is there anything, if there's anything you cannot understand, you can just ask me like a friend, okay? Don't hesitate to question me back. Okay. So they are unicellular. That is their next feature. They are unicellular. Just a single cell. All of them are just a single cell. This all of three, amoeba, paramecium, euglena, they are normally living in aquatic environments like pond water. This euglena is a photosynthetic one. That means that is why it is green in color. It can do photosynthesis to produce their own food like a plant. So it's in just green color ponds. And we can see some times fish tanks become green in color. So if we take a sample of that fish tank, in that water sample contain this kind of euglena like protozoans. And paramecium is normally found in it's,
Rotten hay,
yeah, that is the sample, rotten hay. If we take some hay and put in water for few days, and at the end of that time period, we can observe paramecium in that sample. Really good, you remember that. So that is paramecium. And then amoeba, it is normally in dirty water. That means the water which is mixed with polluted water. Normally, they are found in polluted water, like Bear Lake. They can be found there. Once we were in the university, we are taking samples from Bear Lake to observe these things. Amoeba. So those are the microorganisms belong to this protozoa. They are found in normal aquatic environment. But look at here, this is plasmodium. There's a human body drawn here. That means they are living in us. So plasmodium causes the disease malaria. It is the vector here, the organism which carries that plasmodium to our body is a mosquito. This mosquito bites us and that plasmodium enters into our body and it invades our cells. අපේ cell ඇතුළට එනවා. Then they multiply and then they release out of the cells and they reinfect another cell. They are growing on our cells. So it's really dangerous. It causes disease malaria. So this is the things about protozoans. Did you understand?
yes, teacher.
Yes, okay. So we are moving to next one. My little friends, other ones you have to talk, okay? So, Tisal. Tisal,
Yes, teacher.
Ayan, yeah. Okay. You can talk with me, okay?
Hmm.
Okay.
Okay, teacher.
Okay. So we are moving to next one. Protozoa, and next one is algae. Algae is green in color. When you look at these slides, you can see just they are green in color. Green in color means they carry out photosynthesis because they have green color.
Chlorophyll.
Yeah, good.
Chlorophyll.
Chlorophyll. That is the green color pigment they have. That's why they are green in color, like other normal plant leaves we are seeing. We are seeing our day-to-day life. In here and there, we can see green in color. So this is chlamydomonas. Chlamydomonas. It's just like this. When we draw this, it has a green color cup-shaped chloroplast, which is having green color pigments in it, and it has two tails. Can you remember the names we were using to emphasize these tail-like things?
Flagella.
Flagella, good. They are flagella. Flagella. It has two flagella. It has two flagella to move here and there. And then they can be unicellular or multicellular. This Chlamydomonas is unicellular, just a single cell. But multicellular ones are there. Look at here. This is Spirogyra. Spirogyra. When we draw a Spirogyra filament, it's like this. It has cells, separated cells. Many cells are there, just in a single filament. And it has this type of spiral shape chloroplast. This type of. Here, this image is given here. It's a Spirogyra filament. Its actual, what we can see is given here. In this person's hand, there's a bunch of Spirogyra filaments. They are green in color. Now, you can remember, you may have seen this kind of things in pond water. And in a fish tank, we can see, once we are not washing it for a long time, or we are not giving the proper conditions for that, we can see a little green color filaments are growing on the walls of the fish tank. And those are green color algae. That means there may be different, not only Spirogyra, there are many, but Spirogyra is just one example. And diatom is another one. It's just a box, this kind of box with two flaps. They are diatoms. They are not normally carrying out the photosynthesis, but this image is a little bit green, but they are not doing photosynthesis. Most of them are having chlorophyll for photosynthesis. That is the normal feature of this algae group, but there are some exceptions too. So they are filamentous or thallus body, and they are microscopic. So filamentous or thallus body. Filaments means this kind of Spirogyra. Thallus body means this type of Chlamydomonas, or just like if you have seen in a beach, normally in Galgisa beach, I have seen this one, and or in Gold Face, when you go there, you can see some green color thallus like things are there. On the rocks, you can see some green color thallus-like thing. They are also algae, but they are not microscopic. We can see them as a large thallus. But here, Chlamydomonas, and they are microscopic, even Spirogyra. So we call this unicellular Chlamydomonas like algae as phytoplankton. Phytoplankton. That means in green color water they are living in. The water green become green color due to the presence of this microscopic unicellular green algae. We call them as phytoplankton. This is a good source for the food of little tiny fish. in a pond. Then the macroscopic ones are visible. They can be seen by the naked eye, like this ulva. The ones I mentioned here, this green color one is ulva. It can be seen by naked eye. It's microscopic. Sorry, it's macroscopic. Macroscopic means big. Cannot be, sorry, can be seen with naked eye. Microscopic means small. Then that is about algae. Is that clear for you?
Yes, teacher.
Yes. Okay, then we are moving to our last group of microorganisms, that is viruses. They are somewhat different from other microorganisms. When you look at those microorganisms, they are just cells. They are cells. I told you here there are cells, and Chlamydomonas is a single cell. So in this way, we were talking that they are multicellular or unicellular. But here, they don't have a cellular organization. They are not a cell. This is a image of an coronavirus. This moving one is a coronavirus image. There are some another viruses, HIV, hepatitis B, Ebola. So they are having different shapes. They are not like other normal organisms. Other normal organisms carry out different life processes. We call them as living characteristics, just like respiration, growth, and movement. There are different living characteristics, but they don't show most of the living characteristics. But some of the living characteristics are shown by them, like reproduction. They can reproduce, they can make some new virus particles by their own when they are infected into our cell. This kind, when this coronavirus virus particle enter into our body, they can multiply. They can multiply and become a large number. That's why they can reproduce. That's a living feature, living characteristic. But they are having non-living characteristics too. When we look at the structure of this kind of virus, it's just a protein capsule and just a genetic material molecule in it. It's just like a particle. It's not a real cell. It's just assembly of few different chemical compounds. So they are not available to see under compound microscope in a normal laboratory conditions. They can be seen under a special microscope named as electron microscope. So these are the features of viruses. They are not like normal other microorganisms. Is that okay? Is that understood?
Yes, teacher.
Yes, teacher.
Yes. So we are moving now to discuss about the main part of our lesson. Here we have another one. This thing, I was discussing this thing at the beginning of the lesson, but here there's a new lesson, so I'll tell you that again. These are the environments of microorganisms. In soil, we can find them. In water, we can find them. In atmosphere, we can find them, but only up to 6 km on the atmosphere. There's no more than that limit. Only up to 6 km, 6 kilometers, we can see them. So these are the places, these are the environments they are living in. As I told you, the extreme environments like hot water springs, salt marshes, petrol diesel, even petrol diesel are their environments. We can't even think that that an organism can live in petrol or diesel, but they are living in, microorganisms are living even in volcanoes, deserts, they are living in those environments. So this is about their environment, and now we can see what are their substrates. Here in this image, what do you think about their substrate? Where do they live?   do  they  live?   do  they  live?   do they  live?  do they  live? do they  live? do they  live? do they  live? do they  live? do they  live? do they  live? do they  live? do they  live? do they  live? do they  live? do they  live? do they  live? do they  live? do they  live? do they  live? do they  live? do they  live? do they  live? do they live? do they live? do they live? do they live? do they live? do they live? do they live? do they live? do they live? do they live? do they live? do they live? do they live? do they live? do they live? do they live? do they live? do they live? do they live? do they live? do they live?
They live everywhere.
Yeah, they live everywhere. This person is going to do an hide and seek, do a hide and seek sport, but here they are everywhere. He can't even think. They are everywhere. So they live on or within the body of plants and animals, and also on our food, maybe meat, fish, or vegetable, fruits. They are living on those things. We know that they are causing different diseases to fruits and vegetable. Under this lesson, we are talking about these things too. But here we can learn they are living on different substrates. Not only on our food, they are living on human skin, mouth, anywhere, alimentary canal, urinary tract, everywhere they are living in. Actually, everywhere. And then now we are going to talk about the effects of microorganisms.
Teacher,
Yes,
what is known as alimentary canal?
alimentary canal is our digestive tract. Digestive tract. Yes, in that way you can ask me. Digestive tract is a good place for microorganism to live in. The reason is we are eating food. We are eating different foods. So when the food is polluted with microorganisms, they will enter into our alimentary canal. They enter into that and cause us diseases. In that way, they live everywhere. Okay, good. So here, there are beneficial effects. So when we think about the effects of these microorganisms, they are giving us advantages and disadvantages too. Not only advantages, they're giving us disadvantages too. So here at first, before talking about the disadvantages, we will discuss about the beneficial effects, their uses, their importance in our day-to-day activities. So there are 4 sections we are going to discuss here. These things are, this area where they are important is agriculture, medicine, industries, and bioremediation. Bioremediation, I know that it's a new word for you, so we will discuss that under that section. So here in agriculture, we are moving to discuss about the beneficial effects on agriculture. So these are the importances. Here you can see a golden color rice bottle. So that rice is gold in color. That means nice yellow in color rice. It is named as golden rice. And here you can see it's a
Seameals.
uh, sorry, can you?
Seameals.
Sea maize, yeah. Yes, good. It's a sea maize or a corn crop. And can you see here, there's a little caterpillar which is drawn here. And it is crossed with a red circle. That means that caterpillar cannot eat that corn crop. That crop cannot be eaten by that caterpillar because they are resistant. They are really resistant to that caterpillar. So we call that crops as BT crops. That is the special name we are giving for those crops, BT crops. So this, when that caterpillar eat that crop, its digestive tract or the alimentary canal dissolve because it is toxic for that caterpillar. But normally, normal plants are not like that. Caterpillar can eat easily. But when this modified crop, they are prepared artificially, when it eat this crop, it is destroyed. So they are named as pest resistant crops. Pest, they are resistant to pest. They cannot eat that crop. Not only these maize, sea maize, there are different crops. Mainly not in our country. There are some countries who are growing corn in high scale. And there are different crops they are growing in high scale, in larger scale, in a large amount of acres. just one crop for a large area. So they are using this kind of pest resistant crops. That's a good use of microorganisms in agriculture. So they are prepared by using a special technology named as gene technology. That gene technology uses the bacteria or some fungi to produce this kind of crops. How they do that is they take the genes from those bacteria and fungi. There are some different laboratory methods to cut special genes in those bacteria and fungi. Assume there's a bacteria named Bacillus thuringiensis who is uh, having an ability to produce a toxic compound to kill a caterpillar. Then what these scientists do is they take that special gene from that bacteria and it is uh, it is entered to a plant body by using a special method. That is not mentioned here. It's a special method. So it's somewhat advanced. So we are just remembering that uh, genes can be taken out and they can be used to develop better crops. So this is a pest resistant one and there are some examples here. Drought resistant crops. Drought resistant means those crops can be grown on an area where there is a severe drought. Some areas, even in our country, some areas are really dry. We can't even think how to plant some plants on those areas. But we can develop those plants by entering the genes from different bacteria to develop special crops which has a special ability to grow in those areas. Drought resistant means that areas are very, drought means the areas are very dry. So we can use a gene from a bacterium which is having an ability to live in hot water springs. Then that bacterium may contain special gene to withstand high temperature. So we can use that gene in that crop to grow in that drought or dry area. So did you understand that? How they are made? You all? Is it difficult to understand?
No, teacher.
No. What about other ones?
No, teacher.
No. Yes, Ayan.
No, teacher.
My other students?
No, teacher.
Yeah, okay. So we will discuss other things. Crops with high nutrient. That is this sample, yellow color rice. It is named as golden rice because it is yellow in color and especially it is vitamin A rich. So that gene is taken from a bacterium. That bacterium is Erwinia iridivora. That is Erwinia. E-R-V-I-N-A. Erwinia iridivora. The second name is not need. This is Erwinia. And that bacterium has an ability to produce vitamin A. So that vitamin A producing gene has taken out from that bacterium and entered into this rice plant, the paddy. So now that plant producing rice with high amount of vitamin A. So they are high in nutrient content. And then finally, this is the fourth use, biological pesticides and weedicides. This gene technology can be used to produce biological pesticides and weedicides. That means they are taking the genes from the bacteria or fungi, those which are having an ability to produce different toxic compounds for weeds and for pests. Like here, This Bacillus bacteria, Bacillus thuringiensis bacteria, it produces a toxic compound which is toxic for this caterpillar. It produces toxic compound for this caterpillar, and also we can use some toxic compounds for weeds too. Now chemical weedicides are using in a vast amount in our country, but they are not healthy for us too. Even for our environment, they are not good to use. So in that case, we can use this kind of gene technology applications to produce biological things. They are biological things. They are not harmful to the environment. We are using just organisms. So it is environmental healthy. So but that is one use of agriculture, gene technology. And then next use of microorganisms in agriculture, it is nitrogen fixation. This nitrogen is really important for the plant growth. When your parents are growing anthurium-like plants, flower plants, or sometimes when they are growing vegetable plants, they are buying some fertilizer from a fertilizer shop. So when you look at closely on those fertilizer packs, you can see that they contain NPK. That means nitrogen is the N. PK is another two elements, but you are not learning about those things here. Just I want to tell you that N, nitrogen, is a really required element for the plant growth. That's a very essential nutrient for the plant growth. But this nitrogen is only available in atmosphere. So how can a plant intake that nitrogen? It cannot be taken from the upper plant body through the holes like stomata in plant body. So it should be taken from the roots. But how to take this atmospheric nitrogen from the roots? It is difficult for that plant to do, but they have some friends to do that. They are microorganisms. One example is rhizobium. Yeah, rhizobium. Other one is azotobacter. This rhizobium is living in root nodules. Can you see this image? Can you see these white color bubbles like things?
Yes, teacher.
Yeah, these are the root nodules. These are the root nodules. If you uproot a mimosa plant, අපි කියන්නේ නිදි කුඹා කියනවා. Mimosa plant එක. When you uproot that, you can see nice root nodules like this. But be careful, they are having some tiny spines, but they are having beautiful root nodules like this. These root nodules contain a special bacterium named as rhizobium. This rhizobium live happily in these root nodules because so that plants provide the house for them. Round root nodule. So they live in there and they take nitrogen gas from the atmosphere and convert it to another substance which can be easily absorbed by the plant. That is named as fixation of atmospheric nitrogen. They are fixing it. Earlier, this nitrogen is floating here and there. Now they are fixed. So they are in root nodules of legumes like
Bean, peas.
yeah, bean, peas, and also some they are in other mimosa plant too. Beans, yes, beans, and peas. Yes, in this way, they are living in plants. And there's another group who are free living in soil. They are not in that root nodule. They are just in soil. That is azotobacter. So this bacteria can be used as fertilizer, isn't it? No need to use that artificial chemical fertilizers named as NPK fertilizers and all in fertilizer shops. And but this is available as a biofertilizer, but they may be costly, may be expensive, but it's really good for the environment. We are just using a bacteria and we are adding it to the soil to do this nitrogen fixation process, and it gives the nicely fixed atmospheric nitrogen to the plant. So this biofertilizer, it has a definition here. It's a substance which makes soil rich with nutrient by using microorganisms. So that's a environmental friendly process. So did you understand this use of the bacteria or the microorganisms in nitrogen fixation? Is that
Yes,
fine?
teacher.
Yeah, okay. Uh, you all have to download that tutorial. That means the PDF tube, it's like a short note. You have this entire note nicely. You have to take a color printout. It's really fine if you can take it as a color printout. So you can feel those things. Otherwise, you may feel today, is it boring without writing anything? How do you feel about that? Fine or not?
Fine, teacher.
Fine.
Fine, fine. Do you want a break? Do you want to have a break? Now it's just an hour. You want a 5 minute break? Shall I give you?
Okay, teacher.
Okay, I'm going to give you a 5 minute break.
Okay.
Come soon. Now it's 6. You can come here on 6:05. Okay?
Okay, teacher.
Okay. Now let's start with that after you are coming there. Okay. Okay. Hello? Are you there? Did you came back? Have you come back? Can you hear me? The microphones are still switched off.
Yes, teacher, I can hear you.
Yeah, okay. What about Iron and where is our other students?
They left.
Have they left?
Yes, teacher.
Yes,
Yeah.
miss, I can hear.
Yes, happened to them. They are not coming back now. It's just a break. It's like they have left the class. I couldn't tell them just mute and come back after that. Will need to be take some time to join for them. Oh my god. Hmm. Still not. Now it's becoming 10 minutes. Oh, yes, coming. Yes, they are coming. Yeah, so Indy came back. Ayan is there.
Teacher, really sorry for the delay.
Yeah, it's really fine. I thought that you were disconnected and you couldn't come back. It's fine. Okay. So the name which is mentioned here as Sudin, can you tell me your name again?
Infaz.
Infaz.
Yeah, Tuan Infaz.
Tuan Infaz. Okay. Good. So everyone is here now. Vinuja is also here. So we are moving to our lesson again. Before that, shall I show you something? It's a video about paramecia. It's a nice image. I found that. Give me a second. I'll show you that. It takes time. It takes time. So it won't come easily. So we can move on to our lesson. I'll show you it later on. Otherwise, it's okay. Here we are moving to our presentation again, our lesson. We were here. Beneficial effects. And under the beneficial effects of microorganisms, we are going to talk about another most important fact where we are using microorganisms in agriculture. That is producing compost. Compost fertilizer is produced by using microorganisms. You can't see still. It's just loading. Really sorry for the delay. It takes some time. What happened to that? Yeah, it's coming. While I was going to show you that video, it was like stuck here. Oh. stop share. I'm going to stop the share and come back again. Yes, fine. Finally, it takes some time. Really sorry for the delay. Okay, can you hear me?
Yes, teacher.
Yeah, oh, it's fine. Now this is the third use of microorganisms on agriculture. It is producing compost fertilizer. So compost is produced by decomposing different dead organic matter like plant matter, like animal matter, like animal excretory matter, or maybe different vegetable parts in our kitchen. They are organic matter. So when we are piling up them and we are covering them for few amount of days or little period, we will observe that those organic matter is decomposing little by little. Finally, they become a fertilizer like a soil like mass. So this process is done by microorganisms. So it's a rapid decomposition. It is done rapidly by microorganisms, not just a slow process. It's a really rapid process. It is done by microorganisms like bacteria.
Bacteria, fungi.
Yeah, good. Bacteria and fungi. Even in those kind of compost piles, you can see that fungi filaments like white color little strips වගේ අර threads වගේ නූල් වගේ threads වගේ filaments you can see. So you can see these things. They are bacteria and fungi. We can't see bacteria with naked eye, but we can see fungi filaments. So they are doing this. It makes favorable conditions for plants to grow. It's a fertilizer, and also it's not like a normal fertilizer we are taking from a shop, because that fertilizer is just a chemical. It is just a chemical. But here, this is made up of organic matter, and it make the soil structure and it nourish the soil. And then actually, what happened to this one? It was a real bad thing here. It is just stuck. Yeah, now it's fine. Then fourth use is biopesticides. It is used in agriculture. Biopesticides, as I mentioned earlier in the section of the gene technology, these biopesticides are used for the control of different insect pests who are destroying our crops. If not, to remove the uh, useless plants like salvinia in pond, uh, on the top of the water bodies. That salvinia is not important for a normal pond. That should be removed for the beauty of that pond. So that is done by
Alternaria.
yeah, that is done by alternaria fungus. You can see here brown color dots like things and some filaments. That is alternaria fungus. It grows on aquatic weed salvinia and little by little digest it. So it can control the growth of that salvinia. It prevents the damage to that environment. And also not only for that, here as you can see here, this is the insect name as ladybirds. This eats different insects who are who are damaging our crops. That is another example. So these are really important in agriculture to use them as biopesticides because they are not chemicals, they are not harming our environment, they are not damaging our environment, so they are environmental friendly. So these are the benefits of microorganisms on agriculture. And then we are moving to discuss about the benefits of microorganisms in medicine. There are three uses. We can use them to produce antibiotics, and then we can use them to produce vaccines, and later on we can use them as the organisms to produce antitoxin. So these are the three uses here: antibiotics, antibiotics, vaccines, and antitoxins. Then can you remember those things? Antibiotics, vaccines, and
Antitoxins.
antitoxins. Yeah, these are the three uses of them in medicine. Then antibiotic is a special chemical compound which is produced by a bacteria or a fungi. It is produced by them, that means by microorganisms. They do that, they produce that to kill another bacterium or fungi which harm them. That means when a bacteria coming to destroy a fungal culture, fungi filaments, that fungi produce particular chemical compound that can remove, that can harm, that can attack that bacteria. The first ever found, firstly invented antibiotic was penicillium, which is mentioned here. It was not a thing which was found by research, but just just an accident. So as an accident it was found, but finally these antibiotics become really important in our day-to-day lives because we are taking these amoxicillin, penicillin, and tetracycline like antibiotics to get the protections away from to protect our body from the attacks of
Microorganisms.
bacteria attacks. So they protect our body, antibiotics. And also erythromycin, it is another one. They kills bacteria. And there's another one named as griseofulvin. It can kill fungi.
Fungi.
It affect on fungi. Griseofulvin, that is the one, griseofulvin. So here you can see there's a bacterial cell and antibiotic is coming there that enters into the bacterial cell and it react there, and finally water also entering from that pores and bacterial cell is broken down. The cell ruptured and it destroyed. That is the process done by antibiotics. And then producing vaccines. Now this is very, very popular these days. Vaccines for coronavirus. It's really popular. This is an image of that AstraZeneca vaccine, which is produced to protect us from the coronavirus. So to produce this vaccine, that's a very, very, very it's like it is very it's a very curious process, because here what they do is, during the production of these vaccines, what they do is they take the microbes. That may be a virus. When we think about this coronavirus, they take the virus. Sometimes they weaken the microbes, this virus, by adding different chemicals or different processes, maybe harming them, maybe crushing them, or as an example, they weaken the microbe, or sometimes they kill that microbe, they kill the virus. Then they take the toxins produced by microbes, and then some body parts of that microbes can be taken. So in that way, they are preparing a liquid, or a that means a liquid sample or medication, medicine which contain that virus particles, those which are weakened, killed, or the toxin produced by them or their body parts. So that resembles the disease causing microbe. It's not just the coronavirus, but a part of it. So when it is entering into our body, it is not harmful as the coronavirus, but it can provide us the protection by developing immunity within us. It is coming to our body, develop the immunity. Now we are safe from the actual coronavirus once it is entering into our body. So that is the process of a vaccine. So the examples of weakened microbes are polio, tuberculosis, and measles. These are the diseases. There should be separations here, otherwise it's like just one name. And then cholera, influenza, typhoid, they are the vaccines produced by killed microbes, because they are really dangerous. Then they are not using them as the weakened or body parts, but they are killing the microbe and entering it as a, by inject us it as a vaccine. They are entering that to our body. Then toxins, tetanus and diphtheria. Yeah, diphtheria. Tetanus and diphtheria vaccines are produced by using the toxins produced by those microbes. Maybe a bacterium, maybe a virus. They produce toxins. So those toxins are taken to make a vaccine and enters into our body. So our body thinks then, oh my God, a bacterium or a disease causing bacterium or virus is entering. So we have to provide the protection to this person. So it make different chemical compounds named as antibodies in our body. So they start fighting, and they remember, yeah, there was a little disease causing thing.
Fever.
Hello, did you ask a question?
No, teacher.
Okay, fine. I found that someone is asking, so that's why I asked. So in that way, once the actual disease causing bacterium or virus is coming, our body can identify, yeah, this is something I have known earlier. So it take the guns like things and all the protections. That means antibodies produced in our body and white blood cells, they are protecting us. They come and destroy that particular virus or bacterium. So that is the process of a vaccine. And then hepatitis B is a vaccine produced by the body parts of microbes using genetic engineering. And then, is that fine? Is that understood?
Yes, teacher.
Yes. Now we are moving to last usage of the uh, last application of microorganisms in medicine. What we are discussing here, it is antitoxin. Antitoxin, it's a chemical substance. Actually, it's a toxin which is used to remove toxins produced by pathogenic bacteria. When the pathogenic bacteria is entering, it produces some toxins like this. But these things can harm us. So another microbiome in this environment can produce a antitoxin which can remove and destroy these toxic compounds, these toxins. Bacteria or pathogenic bacteria give us toxins, and we have another antitoxin which is produced by another bacteria or a fungi that is named as an antitoxin. As mentioned here, this tetanus vaccine is produced by using toxins of microbes. So again, this is named as tetanus vaccine. Tetanus vaccine is an example for an antitoxin. So both are coming in the both sections. Under the vaccines it was there, under the antitoxin it was there, because vaccines may be different, but antitoxin is just a toxic compound. Is that clear?
Yes, teacher.
Yes. Okay. And then we are going to discuss about the use of bacteria in use of fungi, like different microorganisms, maybe bacteria, fungi, algae, protozoans, whatever, they can be used in industries. As humans, we are using all the animals and all the plants in this nature. But because we think that we are superior, we are using everything in this nature. So to produce our different things, we are using microorganisms. So the first use is producing biogas. So this is not bad to use them, bad to use these microorganisms in different industries, because they are natural things rather than using toxic chemicals and high heat like conditions. It is really good to use this kind of microorganisms. So first usage is biogas production. Biogas, it is made by anaerobic decomposition of organic matter by anaerobic bacteria. The anaerobic mean they are not using oxygen to respirate. They don't need oxygen. Without oxygen, they can live. So what they do in this biogas production is they make a large pit like this. As in this figure, they are making a large pit. We call that pit as the biogas generator here. This is just a clear image of that. No need to remember all of these words mentioned in this image. Just this excretory matter of this animal is coming here, and they are decomposed by anaerobic bacteria, because this is enclosed area, because there's no entry for any other atmospheric air or anything. It's a closed environment. So there's no oxygen. They can do that process anaerobically. So this anaerobic decomposition produces a special gas named as methane. This methane gas is the source of, we can use that as a fuel. Normally this is done by small farms. What they do is they collect all the excretory matter from the cattle living areas, and then they take all the food matter. If it is a farm with some crops, all the crop waste, all the things are combined, all the things are added to this pit. They are anaerobically digested and methane gas is produced. When they produce methane gas, they can use that for cooking. In that way, they can use that. Then this is done by a special bacterium called methanococcus. It's a round bacterium. It make methane. Since it is making methane, since it is producing methane, it is named as methanococcus. Methanococcus. So that produce the biogas. The main composition of biogas is methane gas. There may be some other gases, but methane gas is the main component of the biogas. Then, did you understand that one? All of you?
Yes, teacher.
Yes, yes, okay. Then I am moving to next one. This is the second industrial application, metal extraction. There are some low-grade ores. That means they are not much rich in soil, rich in different metals. Like as an example, we are extracting iron, Fe, iron from iron containing soil, which is named as hematite. It is rich with iron. This is iron. This is the symbol of iron. Don't think much about this, but this iron is obtained from soil. That soil, that hematite, then soil named as hematite, contain high amount of iron. But there are some elements, uranium, copper-like metals, they are found in very low amount in soil samples. So we call them as low-grade ores. O kiyanne, this word O means the soil with metal. A soil containing, so a soil contained with that, a soil composed with a particular metal is named as ore. Then for this iron containing soil, we call it as iron ore. Iron containing soil is iron ore. So there are some low-grade ores with uranium and copper. They cannot be obtained by different mechanical processes. So what we do is we use microbes who can separate uranium and copper from those soil samples. This process is named as bioleaching. Bioleaching is the extraction of metals from low-grade ores by using microbes. Here you can see there are some copper coils, copper wires. We can see them inside the conducting wires. So this copper is a element sometimes which is less in amount in soil. So it can be obtained by this bioleaching process by microbes. So that is one application. The next one, now we know this properly, dairy products. The most popular one is yogurt. Yogurt is produced by using bacterial processes. Curd. Curd is another thing. Curd is normally produced in household sometimes. But yogurt also can be produced like that once we can buy another yogurt from a shop. So in that way, these dairy products, milk-related products are produced using a special bacterium named as Streptobacillus and Streptococcus and Lactobacillus. Lactobacillus and Streptococcus. So actually the process is given here. What we have to do first is we have to take a milk, maybe a liter of milk. We can take a fresh milk bottle, fresh milk packet from a food city or somewhere. Then we have to boil it up to 88 to 95 degrees of Celsius. We have to boil that. That temperature we can measure that, or if not, that is the temperature we can just put our we can't put our hand. Actually, that is the boiling temperature. Then once we are boiling milk like this, it removes unfavorable bacteria. Again, if there is any microbes here before yogurt production, we kill them, we remove them. Then we add sugar and gelatin to this hot milk. Then sugar and gelatin is well dissolved in this hot milk. And later on, we kept it cool. After cooling to a level where we can put our hand, we are adding a culture of 2 favorable bacteria. So the meaning of this culture of 2 favorable bacteria is we can take the available cultures, or we can just take a previously prepared yogurt from a shop. We can take a yogurt and we can put it into our boiled milk sample, which is dissolved with sugar and gelatin. And then this streptococcus and lactobacillus bacteria in this yogurt, it convert milk into lactic acid. That's why we feel some kind of uh, um, yes, acidic flavor in a milk sample, sorry, in a yogurt or a curd. We mostly feel this flavor in curd, but but yogurt is well flavored with sugar, so we don't feel that kind of taste. It's sweet in taste, but curd gives that taste of lactic acid. Then we have to keep this mixture in separate cups and in a refrigerator to prevent further growth of bacteria. While we are keeping this in refrigerator, the gelatin is solidified, then it makes yogurt. And lactobacillus gives that taste of lactic acid to our dairy products. That is the process of production of yogurt. Is that understood?
Yes, teacher.
Yes. Will you be able to prepare yourself at home with your parents? give it a try. Okay.
Okay, teacher.
Okay. Uh, and also you can prefer your textbook. There's a uh, page. The page number 18. Sorry, page number 8 has that process. You can refer that once you are going to prepare your yogurt. Okay. So that is the next use. We are we are using brooms and some brushes prepared of coconut fibers. These fibers are separated by using microbes. Actually, we can't think about this process if we don't have them. Now there are some machines are prepared for that. Machines are designed for that. But earlier in the area where there's a high amount of coconut cultivation, they are using this coconut husk to prepare fibers. What they do is they put this coconut husk in water to digest uh, that powdery material in between coconut fibers, that is pectate, that is digested by bacteria, because bacteria produce pectinase enzyme. Pectate is digested by pectinase enzyme produced by bacteria. Then fibers are separated. So we can use those fibers to do different products. There are some examples for those plants: coconut, hemp, palmyra, agave. So these kind of plants are used for fiber production. So this is environmental friendly rather than using plastic fibers. So plastic fiber, they are not decaying, biodegrading. So biodegradable fibers can be obtained to produce different clothes, to produce different brushes. So this is really environmental friendly, and this is a good industry based on the microbial decomposition of pectate between fibers. And then this is the last application of microorganism to for the purification of water bodies and for the purification of soil which is polluted with different oils, different toxic material, and also different polluted water removed from the household areas. What they do here is, as an example, you can see here in the top, the upper image, it's oil spills. This blue color one is a ship, and around this ship we can see brown color oil spills. Some oil has leaked out from the ship. So it is really dangerous when oil is floating all over the ocean body. The ocean, the different aquatic organisms are affecting, and also sunlight is not reaching the bottom of the uh, ocean bottom. So it's really disadvantage for the environment. When it is normally, these oil spills are floating to the beaches. When they come there, all the birds and all the aquatic organisms, they are covered with these oil spills. It is really difficult to remove them by using human activities. So we are using microorganisms now. We are adding a microorganism which can digest this soil. Then it is growing on top of these oil samples and digest it. So we can easily remove that. Since they are growing fast, they do this very fast rather than collecting little by human. And the next one is a pond actually. It is filled with wastewater at the beginning. But after putting some microorganisms here, it has become clear at the end. Can you see this? It has
Yes,
become
teacher.
clear. Yeah, now it is nicely blue in color. So this process is named as bioremediation. It's like a medication, but is my bioremediation. The use of microorganisms for the environmental conservation. Environment is protected by using microorganisms. So these are the beneficial effects. Uh, these are the all beneficial effects of microorganisms. Later on, we are going to discuss about the reasons why they are using in different industrial processes, because we are using them to produce yogurt and we are using them to produce coir products, that means the fibers. And then there are some reasons for that. Otherwise, we can use machinery, but we are using microorganisms because they have few small importances for these processes. The most important thing is they are fast growth. They are growing very fast and digesting all the substrates very fast and carry out their digestion process to give us an advantage. And then they act on various substrates, not only one substrate. It may be soil, it may be water, it may be diesel, petrol, like oil spills, anywhere. They can grow on different substrates, not just a single type of bacterium. There may be different microorganisms who can grow in everywhere. So it is another important feature. Then they can be genetically modified. As you see here, in the first application of them in agriculture, in gene technology, they can be genetically modified. They are single genome. When we look at a bacterium, it has a little amount of genetic material, not like us. A single small DNA molecule. We can cut it and take out easily. The required gene can be added to another organism. So this is named as the ability of them to genetically modify it. They can be modified. And then microorganisms are available for low cost, if not free of charge. They can be available, they can be taken at a low cost. Sometimes they are free of charge. In the process of vinegar production, we don't have to find them from the shops. If we keep a coconut sample of coconut water somewhere, a bottle of coconut water somewhere, it freely turns into vinegar at the end. And also, for the yogurt production, we don't have to buy microbes at a high cost. We can just take a yogurt and prepare that yogurt. And then they need less energy. Less energy is required for them. For the machineries, we know that we have to use more fuel. We need fuel. If not, we need much electricity. But for microorganisms? It is not a necessity. They can easily grown without any special requirements. And then finally, another special advantage, another special feature of these microbes to use them in industry is minimal environmental damage. As I uh, told you earlier, when I was discussing all the applications of these bacterium, a bacterium or fungi or any other microorganisms, I was telling you that this is environmental friendly. They are organisms. They are not harming environment. So that is the special feature. They have minimal environmental damage. But we know that other different chemical fertilizers, they are harming the environment. Different weedicides, chemical weedicide, different chemical pesticides, they are polluting the environment. But microorganisms are not like that. So we can use biofertilizers instead of chemical fertilizers. So these are the special features that why they are important in large scale production. So these are the all things about the uses of microorganisms in different applications. That means benefits. They are benefits. Do you have something to ask from me about their beneficial effects or their beneficial uses?
No, teacher.
No.
No, teacher.
Okay. Then shall we move to their disadvantages, their harmful effects?
Okay, teacher, let's
Okay.
move.
Okay. So then start. It's not moving. It's not going to the next slide. Give me a second. It's stuck. Yeah, fine. Okay. So here we have another only five more slides. They are about their adverse effects or their uh, disadvantages on us. First of all, they cause diseases. We know that it's a disadvantage. They are causing diseases to humans and plants. And later on, they are carrying out the food spoilage. They spoil our food. Finally, they can be used as biological weapons. Those are the adverse effects of microorganisms. So let's discuss about the process of causing diseases to humans. Here, we have three special terms to remember. Can you read them with this green color words here?
Pathogen, vector, host.
Yeah. What about other students?
Pathogen, vector, host.
Vector, host. Yeah, pathogen, vector, host. So pathogen is the microorganism with a potential to cause a disease. There may be bacteria, there may be different viruses, then sometimes they may be fungi. We know that fungi also causing diseases to human. And then they are the pathogens. They cause diseases to us. They have a potential, they have an ability to cause a disease. And then there's a method for them to reach the host. that there may be a particular organism which carry those pathogens to the host. That is named as the vector. The most common vector we know is
Mosquitoes.
mosquito. Yeah, mosquito. They cause. Have you have an idea what are the diseases caused by mosquitoes?
Yes.
Yeah. Can you tell me just few examples?
Dengue, filaria.
Yeah, dengue and malaria also. All the things, these kind of diseases are taken, these disease causing pathogens are carried to us by these vectors. There may be different vectors. And sand flies, they are also carrying those um, pathogen to us. And then host is the organism who provides this inside or outside body as a substrate. He is the person or the organism who get the disease due to the pathogen at the end. So these are some examples of diseases caused by pathogen to human. There are different pathogens, bacteria, viruses, and protozoa. And diseases are, the bacteria causes tuberculosis and typhoid fever. Viruses are causing
Cold,
cold,
dengue,
yeah,
HIV.
dengue and HIV.
HIV.
Yeah, they are caused by viruses. Then these protozoa, what are those?
Malaria,
Yeah,
amoebic
yeah.
dysentery, leishmaniasis.
Three, leishmaniasis, malaria, amoebic dysentery, and leishmaniasis. These are caused by protozoans we have learned. And then these are the diseases caused by microorganisms to plants. This is powdery mildew. When you look at a crop, it may have a little white color, powdery, this like threads-like substances on a crop. Normally, chili plants are having this. You may have some chili plants on your garden. So you can see sometimes, if we are not uh, looking at them carefully, we can't see, but at the bottom of the leaf we can see sometimes powdery substance that is named as powdery mildew. Then sometimes wilting occur due to different microorganisms. That means the
Fungi.
microorganisms like fungi and bacteria, they are entering into the xylem vessels in plants. When we look at a plant cross section, we can see that xylem vessels are along the stem. So these are the vessels who carries water to the leaves from the roots. When these are blocked with bacteria or different fungi, water cannot pass through these xylem vessels. These are the xylem vessels.
Tissue.
Xylem tissue. Yeah, xylem tissue. It is the tissue which transport water in plant body. It is the tissue which transport water from roots to the leaves. Then when they are blocked, no water for the leaves. Although they are green in color, they wilting at once. They wilt at once due to the lack of water. And then next disease is potato late blight. Potato tubers and potato plants are affected with this. They become black in color, and black brown spots can be seen. It occurs due to a fungus. Fungi cause this potato late blight. Later, these brown spots are turning into black. Then this disease damage all the harvest of potato. So these are the uh, diseases caused by microorganisms to the plants. Here, powdery mildew is again, it is caused by a fungi. Fungi is participating to cause powdery mildew, and potato leaf blight. Wilting is caused by both bacteria and fungi. Both are participating to cause this wilting of the plants. So then the next disadvantage of them is food spoilage. We know that we are um, having this experience in our day-to-day life. Bread like things, they become spoiled. When the sugar containing foods are spoiled, we call that process as fermentation. When the sugar containing foods are spoiling, it is fermentation. When protein containing foods are spoiling, we call that as putrefaction for protein containing food. And here, this is fat or lipid containing food. When they become spoiled, we call it as rancidity. So these are the three methods of food spoilage. The reason for this food spoilage is growth of microorganisms. They are growing on these foods. So we can see here this brown color, black color spots. Here this black color, white color spots on bread. They are microorganisms. So they grow on food. And then they release toxic material. Due to the release of toxic material, we feel a change of flavor. Flavor is changing due to their toxic material. So we can define this food spoilage as the change of chemical and physical. Chemical composition is changing and physical nature also changing. Physical nature is what we see. And what we see and what we feel actually. When we touch a spoiled bread sample, it is just really rough and it is not smooth as a good bread sample. And then this is the last adverse effect caused by microorganisms. So that is the use of them as biological weapons to destroy human in war activities on military purposes they are using. The most common example of use is the use of anthrax disease. This is not moving to the next slide. Give me a second. Anthrax disease is caused by anthrax is bacteria, Bacillus anthracis bacterium. So it gives this disease. This pathogenic, harmful bacteria used in military purposes. Once they reach the victim, they are causing the diseases. So no need to use another weapons. All the community is infected with these diseases and they are damaged. So this is the normal thing we have to discuss in this lesson. Have you understand? Have you understood? Did you understood everything I have discussed here?
Yes, teacher.
Yes. Do anyone have any problem? You can ask from me, just like a friend, you can just chat with me and ask anything from me. What about everyone? I'm going to stop this presentation here. And then, so is there any problem? Is there anything for you to ask from me? There's a message from Savindi. And here, at the end of the classroom, yeah. So I have to tell you that. So I'm going to tell you that in that website, in that platform, you have your profile, your uh, student profile. In that, you can, so I show it. Give me a second, I'll show it you. Here. Once you have answered the question papers and all the things, you can submit. You have an option upload. So this is a teacher profile. In the same way, in your profile, in the tutorial section or in paper section or in MCQ section, you have a place to upload your answer sheets and everything. So in this tutorial section, you have the grade 9 lesson 1 PDF dude. It's your note. You have to download it and take a printout and put your make your note properly on this. And then later on, at the end of this class, I'm giving you a paper to answer. So you can submit it to this platform. And then if I am giving MCQs, I am giving it in the form of Google Forms, you can upload that also to this platform by using your student profile. Savindi and all, is that clear?
Yes, teacher.
Yes. Do you have any, if you have any problem, you can call to this number. Can you write down that? If you have any difficulty with this platform, you can call to this number. You can give a call. Shall I read it? Can you note
Okay,
down?
teacher.
Yes. 076 571 not 93 not. Did you write it? Did you note that? Did you note it down?
Can you tell it again, teacher?
076 571 0930.
Okay, teacher.
Okay. You can give a call and ask for any help with this process of the uploading your answer sheets and everything. Okay, then shall we stop?
Okay, teacher.
Okay, okay. Okay, remember to take the tutorial and prepare a note before being more late, okay? Okay, goodbye.